Okay, why don't we reconvene? Needless to say, I think NHGRI, I've come to the conclusion in recent years that we're sort of at that stage in our existence where we have lots of great programs going on, but also way too many good opportunities also to contemplate going forward. And a responsible thing to do, especially with budgets that are limited and sometimes are strained, is just to make sure we are properly examining our existing programs, especially ones that have been around a long time, and try to assess them so we could get a sense of making decisions about them and others, and also for setting priorities, because we really are in a situation where prioritization is going to become increasingly challenging. I would also say, and I think Chris Gunter, our presenter, is going to make some comments about this, that we're expected to do this as good stewards of the public's money. And there's even, I think, greater and greater attention from higher up within the administration, the department, and even at NIH, to make sure that institutes are taking critical looks at their programs from time to time and just not assuming everything is equally good and everything should just continue without any changes. And so one of the things that when Chris Gunter joined the Institute a number of years ago, this was even before the pandemic, when we sort of came up with a set of things that would be exquisitely helpful to me as a senior advisor, would be to take some of her research skills and apply them to do some critical evaluations of some of our programs. And that doesn't mean only extramural programs. It doesn't mean other parts of the institute, anywhere where we think there's something that is worth evaluating. And so some of this got a little slowed down because of the pandemic, but now she's here to report on really the completion of the first such effort. There will be other efforts, I can tell you, where we're just trying to develop that expertise internally. And I think Chris has done a great job of using the first example as a pilot to get a lot of experience and also to sort of help us build up some expertise in this kind of evaluation mode. So with that introduction, I'm going to turn it over to Chris Gunter.
Thank you very much, Eric. I am glad to be here sharing the results of our first evaluation with you. And as soon as I can get my slides to advance. Oh, I'm sorry. I want to make sure I acknowledge Molly Bird and Maya Van Zanten, our colleagues who are here from NHGRI, and also a number of colleagues from Ripple Effect as well. So in addition to what Eric said, I want to also make clear that there are a number of different efforts across NIH to establish more formal program evaluations as being required for multiple levels above us. And in addition to what Eric said, this is something that we'd really like to have the data for as well. We're increasingly asked to show evidence for impact, effects of policies, grant spending, et cetera. So that's one of the reasons behind this. We also, as you may know, are very committed to transparency and program assessment. So for this evaluation, we're going to both put it out in a public format, which is a brochure that we worked with Daryl Leha on, which you'll be getting within a week of council. We also, even though program evaluations are different than other forms of research, we also did get IRB clearance, and we do plan to publish this in a peer-reviewed journal. So for our first big mixed methods program evaluation, we decided to look at our quote-unquote flagship grant mechanism, as you all are very familiar with, the Centers of Excellence in Genomic Sciences, or CEGS. Just to recap, that's a P50 or an RM1 mechanism over its lifetime. It's high risk, high reward explicitly. It has teams of multiple investigators who can be at the same or multiple institutions. The focus is supposed to be on transformative approaches and outputs. There are requirements for education and outreach elements, as well as spending money on developing tools, methods, and resources, and sharing those with the community. So when you do a program evaluation, you use something called a logic model, and we won't spend a lot of time on that in the interest of time, but here is our logic model. We had participants in there, literally who participated. We looked at the inputs, what we put in. We measured our outputs, what we did, and how we're going to evaluate those. And then finally, we looked at our outcomes, which are short, medium, and long term. So in the interest of getting all this through in 20 minutes to you in council instead of a 150-page report, which we also have, what I'm going to do instead is focus on 4 main evaluation questions. So the first one is, what did the CEGS program achieve during its lifespan as a result of its key activities? How did this program influence genomic science knowledge and utilization of genomics research? How did this program funding influence grantees' careers? And then what have been the strengths of the program, and do we have any suggestions or opportunities for improvement that came up in the evaluation? So we used 3 different methods, which I'm going to walk you through. The first is that I conducted semi-structured interviews with 28 of the contact PIs for CEGS over the lifespan, again, for the past 20 years, and did coding and thematic analysis. You'll see those appear here primarily in the form of quotes that came out of the interviews and some themes that we got from them. The second method we used was quantitative output, so I'm going to spend a few minutes telling you in more depth about those. So in order to be able to do a comparison, that's kind of difficult for a grant mechanism like this, because what do you compare it to? What we did was take a cue from an analysis that NIAID had done and put together a basket of R01 grants, because those are also investigator-initiated scientific grants. So you can see our criteria here on the left. These had to be funded by NHGRI. I selected them to be similar topic areas. We looked for total direct funding within 5 to 10% of CEGS funding each year, and I'll show you that in a second. Over the lifespan of the grant, we excluded any R01s that were done by CEGS PIs, so they wouldn't be counted, to not be double counted. And you can see the table here on the right showing you we, of course, had many more CEGS, many more R01s to get to the level of the CEGS grants, many more PIs. The average direct funding amount is obviously quite different between an average CEGS and an R01, and the grant length, to keep in mind, is also different, because NHGRI, for the first number of years it was here, did 3 years as a standard R01. And this is the graph I mentioned. You can see that we did fairly well being able to measure the R01 pool versus the CEGS in terms of matching direct funding. There is a blip that you can see from 2009 to 2011, and those of you who've been around a while will remember the American Recovery and Reinvestment Act, or ARRA. That is the explanation for that change there between the two. So what did we do? We looked at a number of different metrics of those, which I'm going to walk you through later. So we looked at metrics of publications, numbers produced, citations, impact measures. We looked at a number of patent measures, and then also grants, applications, and awards, and direct funding. And then our final method was to do a survey where we got both quantitative and qualitative data from CEGS researchers. We extended this opportunity to everyone that we could find who had been on CEGS except the contact PIs, so other PIs all the way down to summer students if we could find them. And just to show you our final demographics, we extended this invite to 230 personnel. We got 62 participants, so 27% response rate. The most common demographic categories were male, white, and postdoctoral fellows during their time on the CEGS grants. So to keep in mind while you're interpreting this, we also have a recency bias, so fully 50% of our respondents are current, basically, CEGS personnel, 42% in the second decade of CEGS, and only 8% in the first decade of CEGS. So keep that in mind. So then moving to our evaluation questions, what did this program achieve as a result of its activities? The first thing we found was that CEGS labs have contributed to genomics advancements in all of the 4 areas specifically targeted previously by the CEGS mechanism as expelled out in previous program announcements. So new technologies, methods, resources, and an understanding of problems. And how do we see that? Well, we asked the researchers, how do you feel that your lab did in contributing to these 4 areas while you were part of the lab? And you can see that graphed out here on 4 categories, and then in each block there are the answers that were not at all or to a small extent in dark. The medium color is to a moderate extent, and the light color is to a large or very large extent. So the majority for each of these said they felt to a large or very large extent their lab had contributed to that category. We also did our own research, and in interviews asked the PIs what they felt CEGS funding had contributed to, and got this list of breakthrough techniques, resources, and approaches. I'm going to come back and focus a little more specifically on RNA-seq and single-cell ATAC-seq later in the presentation. And then finally, when we talked to investigators here, we asked them what they thought the biggest success of their grants was, and they pointed to a number of different areas. And here we have an illustrative quote at the bottom. I feel like the high risk, high reward aspect of CEGS and just the general vibe it sends out is one of the crown jewels in the NIH system. But here are the answers, the themes that they gave us based on their interviews. We heard a lot of new resources, biological questions, linking biology to technology, but we also heard freedom to take risk and difficulty or challenge as one of the big success of their CEGS grants, and a lot of focus on people and training or collaborations or education. So the second question, how did this program influence genomic science knowledge and utilization of genomics research? You can imagine we're going to do a lot of quantitative data here. So the CEGS grants produced more papers and more highly cited papers per grant than the comparison R01s, but fewer papers per $1 million of investment. That's likely due in part to the additional requirements for the infrastructure, such as education and outreach for the CEGS. So how do we see that? We looked specifically at publication metrics. The second column is the CEGS grants. The third is the comparison grants. You can see, as you might imagine, many more publications coming from a much larger basket of grants for the R01s, but more publications per grant coming out of the CEGS. They're multi-investigator, so that's not surprising. We also see an average cost of publication is higher from the CEGS. Again, keep in mind the infrastructure requirements, and the publications per $1 million as a result are fewer from the CEGS. And the graph here on the right is just showing you how that worked out over time. But then when we looked at measures of impact, what we saw using a couple of different measures, the first one is the relative citation ratio, or the RCR. That is a normalized metric developed by NIH, where NIH-funded publications are normalized to a value of 1. So you can see the CEGS grants publications have a median RCR of 1.6, meaning they are 60% more cited than other NIH-funded publications, compared to the comparison group, which is 1.2. The NIH percentile, so taking that RCR and normalizing it on a scale of 0 to 100, is 68% for CEGS versus 57% for comparison grants, and that's graphed out on the right in terms of quintiles for the two groups. We also looked at citation lag, which is the gap between the publication of an article and its first citation. You can see that's shorter for the CEGS, so their papers are cited faster. We also, using the more traditional measure of journal impact factor, which is the number of citations for an article in the past 2 years, that is higher for CEGS grants publications and in a higher percentage if you normalize that from 0 to 100. And then looking again at utilization of genomics research, we found that the CEGS grants were 14 times more likely to produce a patent, which I'll show you. We also, when we talked to scientists, found that they praised the contribution of the CEGS as going beyond quote-unquote standard genomic science and moving into transformative approaches and discoveries. And we also are going to show you that methods developed within these grants have gone on to be used in grant applications that study a number of conditions and are funded by a number of different areas of NIH. So here are the patent data. What we saw, again, highlighted in yellow, 14 times more likely for CEGS grants to produce patents than comparison grants. 65% of CEGS grants have at least one patent compared to 16% of comparison grants. And then some of the PIs specifically called this out as an important area. And you can see the first one is saying with an R01, you don't think about what the problem is in front of the community, but the CEGS basically ask you to solve that problem in front of the community. And the second saying, I think that the CEGS has been a huge boon to genomic science, and that's the kind of funding I find most useful, where you can swing for the fences. You might not always hit things, but we feel like if you do, you're going to make interesting discoveries. So that's what this person's feeling was. And then finally, some slides here generated by Molly Bird, our Policy and Evaluation Fellow from AAAS. So we looked at two techniques, RNA-seq and single-cell attack-seq, and we're going to show you 3 different measures of them being used over time. So the first is looking at NIH-funded awards that mention RNA-seq, and you can see from when it started, in part due to CEGS funding in 2008, to the current year, 2023, it's now up to being used in almost 4,000 different funded NIH awards. We see a similar trajectory for single-cell attack-seq, and the acronym is spelled out there on the bottom. Again, looking at awards, again, that technique at least partially funded by a CEGS, now from its publication in 2014 to being used in almost 800 awards in 2023. But we also wanted to ask, has this technique gone on to be used in a number of different fields? So I'm going to show you again 2 graphs on that. The first is RNA-seq. And what we're showing here is NIH awarded type 1 and type 2 grants that mention using RNA-seq. And this is a time scale. So on the left, it goes from 2008 to 2023 on the right. And the width of each of the bars going across the time scale is how many awards use, in this case specifically on the y-axis, the human condition that is mentioned there. So the top 3 you might be able to see are cancer and neoplasms and inflammation. But the point I want you to get is that there are at least 40 different conditions that are mentioned here. So it's being used in a number of different conditions. And then single-cell attack-seq, we see a smaller, more compressed timeline, 2014 to 2023. But again, cancer, neoplasms, inflammation, the top 3, and being used to study a number of different human conditions. And then finally, we wanted to ask, did NHGRI's investment turn into something that has now been funded by a number of different institutes and centers across NIH? Similar timescale here, but the question that we're asking is who is funding these grants using RNA-seq? And you can see by the width of the bars becoming much more wide over time, that is spread out to a number, in fact, almost all the institutes and centers of NIH for RNA-seq. And then single-cell attack-seq is on a trajectory as well to be funded by a number of different institutes and centers across NIH. So 2 more questions. The third one is, how did this program influence grantees' careers? We saw that the impact of the CEGS program had an impact on careers by improving grant funding levels, and I'll show you that in a second, but also it allowed personnel at many levels to create or participate in outreach activities. So we asked in our survey, did you participate in these outreach activities, which are a part of the CEGS? And what we found is that, yes, up to 71% of the respondents said they participated in summer programs, which would not have existed except for the CEGS, and all the way down to science communication to non-academic audiences, which of course I love. So that was great for us to see that people participated in some of these things. And the second thing is our NIH grant metrics. So let me set this up a little. So our colleagues calculated yearly averages for each group that we looked at by dividing per PI statistics by the number of years each PI had to compete for subsequent grants, starting 2 years after the grantee was a contact PI for either the CEGS or the comparison grants. So that was an attempt to account for varying amounts of time to apply for grants after receiving the grant in question that we're measuring against, where they were the contact PI. And we discussed this a little earlier in council. These numbers are not adjusted for inflation, the funding numbers. So some of these findings could be partially attributed to inflation and also pay lines changing over time. So to look at this, what we saw for the CEGS grantees is that in the post-grant period, they are putting in more applications and more R01 applications. They're not necessarily getting more awards, but they are certainly getting more direct funding when they do get awards. And then we also looked at comparison grantees. We see a similar pattern. We see more applications overall and more R01 applications. We don't necessarily see more awards over time on a yearly basis. Remember, this is normalized to yearly basis. But we are also seeing more direct funding when they do get awards. And then finally, to end, what was our fourth question? Looking at strengths and then opportunities for improvement. So a couple of slides of strengths here. We heard from the investigators fostering collaborations because it's a team effort, supporting high risk, high reward research. And here are 2 quotes about that. An investigator said, "We developed a new method. We unfortunately also found out that it had limitations, but this would never have happened except for the CEGS." And the second one was an investigator saying, in their opinion, if the program's doing what it's supposed to do, you'll walk out of the CEGS meeting and the annual meeting each year feeling like a quarter to a third of the stuff isn't going to work. They also praised providing freedom and flexibility in their research and enabling technology development in an NIH grant. So this is a quote saying, when we look around, there's not too many NIH institutes and centers who are willing to do this kind of tech development. This mechanism is one of the few that's doing that. Okay. We also heard a number of suggestions or opportunities for improvements. The first one is that survey participants in particular mentioned a need for more workshop and training opportunities beyond what's already there, as well as more targeted outreach of diverse populations. We also heard investigators quite strongly felt that NHGRI should ensure that the CEGS goes beyond nucleic acid. So here's an illustrative quote of that. I think that NHGRI has drawn hard lines of whether, if it's not nucleic acid space, it's no longer within the purview of NHGRI. That's been a disservice to genome biology. And in fact, we haven't learned as much as we could have from genome biology, in this person's opinion, based on that. So this is not going to surprise anyone. Another big concern of the investigators was that the flat budgets over time are becoming a problem for them accomplishing parts of the grants. So 2 illustrative quotes here. I'm worried that if we don't have enough resources, we won't be able to deliver, particularly on education and outreach. And the second one being, they wrote their renewal fairly recently, and they worry that they can't meet the mandate of the CEGS because the budget is too small. So that's not going to surprise anyone. And then the final theme that we saw was a number of investigators suggesting that NHGRI consider a shorter or a smaller CEGS mechanism. And you can see that here in the 2 quotes. Maybe they could be shorter. They're only 2-3 years. Maybe there are only 2 or 3 investigators. They're a smaller amount of money, but these are wild. I had to redact the actual word there. Out of left field ideas that were suggested. And then another one was, I think there's some miniature version of the CEGS that's a little more feasible, having more of them and letting more people engage in that high risk, high reward research. I think that would be an interesting concept to be addressed. So I want to thank everyone who was a part of this, in particular everyone at NHGRI and Ripple Effect that helped us, and the survey and interview participants. And then we'll take questions here.
Questions from council? Too much information?
I'd be curious if council members found any of this surprising. I mean, is there anything that surprised you, or especially folks who have been close to NHGRI's portfolio for a long time? Is this pretty much what you expected? Nancy?
I certainly expected the impact. I mean, I saw it. It was hard to miss the impact of those programs. But I do wonder whether some might wonder with the way technology is evolving that the opportunity to renew those maybe needs a second thought. I don't know. And I wonder if it would be useful to look at separately the returns on the investment in the last 5 years relative to the first 5 for those who've actually been renewed. And whether, I mean, it just might be one more thing that gets thought about. I suspect there might be cohort effects in the impact because, really being at the beginning of genome sequencing probably had a good long tail for impact. But it's impossible to know, what the people working on RNAs, the sort of later downstream things that will have, how that will have reached through. But overall, that was a really nice presentation. That was kind of a textbook look at a program, and I think that rubric will stand you well going forward.
Yeah, thank you so much. And I don't know if Eric wants to add anything there, but yeah, I agree with you. There is definitely a time factor here as well. So that's something for us to think about going forward.
Judy?
I was fascinated by your metric of citation lag, and I was thinking 111 days or whatever that number was is actually quite short. Did you stratify by whether they use preprint services, and how do you anticipate using this in the future with kind of the changes in the data sharing policies?
That is a great question. I am fairly certain that preprint services would not have been included in there because the data were pulled from PubMed. So great question, but that's something that we can dig into a little further. And I agree, that's something that we should think about doing in the future is adding preprints in there. I also thought that was quite short, so it was impressive.
Lynn?
Yeah, thanks, Chris. That was a great presentation. Lots of data, and we love data. One thing, maybe I didn't get it right, but was the response rate 27%?
Yes, that's correct. Yeah.
So should we be concerned about possible biases then?
Oh,
with
sure.
that
Yeah,
level?
absolutely. I mean, I think that's the case. And in particular, that's why I wanted to show the graph of the recency bias. I mean, I had to literally track down earlier trainees on the CEGS by trying to use LinkedIn and Google, et cetera. So people moved and are on their 12th job by now. So absolutely, I think there are definitely biases to be taken into account. And that's something for us to think about when we're embarking on our next program evaluation. Yeah,
Thank
good
you.
point.
Peter?
I think there's probably a bias in the publication lag statistic because if you have a big group, they're likely to cite each other. I don't know if you took that into account. And it's probably also the case that there's just more of a network and more communication. And so it might not be a sign that the paper is so impactful that it gets cited right away. I mean, it could be, but the statistic doesn't prove it.
Yeah, no, and that's a great question. We actually do have data removing self-citations from there. They're similar, and just in the interest of time, I didn't want to get into that, but we do have data with the self-citations removed, and the trend is similar. Thank you.
Other comments or questions? All right, Chris, thank you
Thank you.
very much.